All right, so let's get started. Welcome everyone to the first installment of the GHGA webinar. Today we're gonna have a beginner's guide to DNA and sequencing for you. Since this is the first installment, I wanted to briefly introduce what GHGA is to you. It's the German Human Genome-Pheome Archive, and our mission is to provide a basis for GDPR-compliant human omics data sharing, which will enable the secondary use of clinical omics data for biological discovery and replication of findings. And this will involve interactions with very diverse communities like biologists, geneticists, scientists, physicians, and patients, etc. And for more detail, stay tuned for Nick's talk. Speaking of Nick, he is our speaker today, and just as a brief introduction, Nick has a Bachelor of Science in Computer Science with a focus on bioinformatics from UC San Diego, and a Master's in Informatics from the Technical University in Munich. And he's been working for GHGA as a bioinformatician since January 2021, where he works in workflows and data hub operations.
And so I want to welcome you to my presentation. I'm Nick, and I'll be presenting an overview of some basic biology, as well as some introductory bioinformatics. And after an intermission or Q&A, we'll have a quick look into what GHGA sets out to be and what fair workflows can mean for us. I'll get into the specifics of that later, but a quick outline of the two sections. First, we'll be starting out with the biology and eventually getting into a bigger picture of GHGA. So starting with the basics of biology, the simplest model we have is that DNA makes RNA, RNA makes proteins, and proteins do stuff. A bit more detail into that is that the biological information for an organism, and we're going to focus on humans today, so the information for everything that makes us human is encoded into our DNA. That information is transcribed or written into RNA. This is the messenger molecule that carries instructions that are important locally for the cell and smaller subsections. And then eventually RNA is translated into proteins, which are the molecules that interact with each other and allow the cell to interact with the environment and other cells. So a little bit of the basics of DNA, in case you have forgotten everything from high school biology, is DNA has a double helix shape. This double helix refers to the two strands of a DNA molecule. So you can see here, there are two strands in blue, connected by bridges of nucleotide bases. These are adenine, thymine, cytosine, and guanine, and these molecules interact to form bonds across the backbone of the double helix. These can be represented by the letters A, T, C, and G. And note that these bases form specific special bonds. So A and T are always bonding together, and G and C bond together. In fact, you look at this picture, you see red and green always paired together, and blue and yellow always paired together, but not A and C or G and T. It's specific. The human genome is 3 billion bases long and are organized into structures called chromosomes. There are 23 pairs of these, totaling 46 chromosomes. And for each pair of chromosomes, we inherit one copy from one parent and the other copy from the other. The DNA is further organized into two very much umbrella general categories of genes, which make up 1 to 2% of the human genome, and non-genes would take up 98 to 99% of the human genome. And when I say these stats and these figures, all of this is with an asterisk. Everything is more complicated. There's always an exception to every rule. So we'll get into a little bit more of the breakdown. The human genome is really long. Like I said, it's about 3 billion bases, and stretched out in just a single cell's worth of DNA, the DNA stretched out would measure about 3 meters, which obviously is way too big for a single cell. And so it's wrapped around not only itself, but other structures. And these structures are wrapped against other structures, forming these incredibly dense chromosomes, which you can see in this picture from a microscope image. Each chromosome is further broken into genes. There are about 20,000 in the human genome, and each of us have 2 copies of each gene, inherited one from one parent and the other from another. Genes are important because they are sections of DNA that sequence, sections of the DNA sequence that encode for information about proteins. But most of the genetic information in the human body is not encoding proteins. In general, this information between genes is regulatory, either specifying special instructions for how to use nearby genes, or is a physical spacer between genes. So everything I talk about today, remember that all of this is driven by physical and chemical interactions and can be quite complicated. So within chromosomes, we have genes, and within genes, we have more substructures. We'll get into this later, but they're further broken down into exons, which we can see here in blue, and introns, which we see in red. Note that generally, genes excise or remove the red introns to give you the RNA that is only blue, only exons and only coding regions. Again, tons of asterisks. So when we talk about DNA sequencing, our goal is to identify the order of nucleotides, the order of A's, C's, T's, and G's within a genome. So there are around 3 billion, which is a lot of letters to read. And so, in fact, our machines and our sequencers can only read smaller sections called reads. These sections of genetic material can be read in a single go, around 150 to 200 bases long, generally, and represent a random section of the genome that it want to sequence. So the goal is that if you have enough of these reads, you can expect to see coverage across the entire genome, with each read providing evidence and a way to check if the sequencing run was successful. So a sequencing run can generate a certain number of reads, depending on what you'd like to do, anywhere from around 5 million for precise or focused experiments to more than 1 billion reads for large, in-depth experiments. The larger the number of reads and the length of each read will give an estimate of the overlap between them and the coverage at a single location. So when we talk about coverage, we're talking about the sequencing depth. This is how many reads cover a single spot in the genome, or on average across the entire genome. This is evidence that the sequencing is reliable, or that you can find or you can trust that the results you get. So if, for example, you have 30 reads covering a single spot, and 29 of them say that you should have a C, and only one says it's a G, you have a lot more evidence that the C is the correct nucleotide. However, if you have 15 Cs and 15 Gs, then it's likely that you have a split between which one you inherited from each parent. So in this case, you could be potentially heterozygous, where one parent donated a C and the other donated a G. So we talk all of this in context of a reference genome. This is the quote unquote gold standard comparison tool. And the human genome is really just a particular genome or a particular sample that we have studied a lot. Because 99.9% of humans all have the same genetic information, we can use this reference to help figure out where a read should go, and it's essentially a guide for bioinformatic tools. So when we talk about the reads and our reference, the goal is to figure out where they go. And so we align them to the reference genome. And because we have so many reads, we can use the reference as a guide, very much like the picture on a jigsaw puzzle, to see how we can stack up each read in a certain way and in a certain order that gives us, in the whole, a bigger understanding of the genome. So an example of this would be a reference of 10 bases and 10 reads of 5 bases long. And so they're taken from random sections of this, but what we can do is we can order them and rearrange them such that not only do we cover the entire reference genome, in this case, all 10 letters, but we also have different levels of support for different regions of our reference. So for example, we can see that at position 3, we have a lot more evidence supporting this A than we do at position 9, where we only have 3 reads. And at the end, we only have the 1 in this example. So the goal is to figure out where our reads in our sample go and how they differ from the reference. Some of these changes are benign and have no effect. In fact, compared to the reference, most people have around 4 to 5 million variants 4 to 5 million differences between our genome and the reference. And however, that doesn't mean that we all have some rare, crazy genetic disease. And there's a lot of mechanisms to tolerate variants, even when they are in important regions. So let's simplify the project and the problem. So as a computer scientist, it's really hard to model molecules. Instead, I'm going to just focus on the letters themselves. So instead of this double helix and this very complicated picture in front of you, we can simplify it so that different molecules are given different letters in a string. So each red and blue block can be an A or a T, and green and gold blocks can be a C or a G. So the simplified version, at every position from one parent or the other, we can identify the nucleotide at that position. And because we know that this A and T pairing and G and C pairing, we can actually remove half of the information without losing any actual knowledge. So at any one of these positions, we could gather what the other side would be based on what we see here. So at this position, we have an A, and at this position, we would have a T. So we don't actually lose any information, but we reduce our data complexity. In fact, we can reduce it even further by using the reference genome. Like I said, the reference genome is a highly studied individual. It doesn't mean this is healthy DNA or this is what is right. It's just how it looks for this particular sample. So as a result, many parts of the reference overlap with our individual. In fact, most of it is identical. There's only a couple of places where it is different. And these are the regions that we can immediately focus on. So in positions 2, 7, and 10, we see that there's one difference where we have inherited one difference compared to the reference. In position 3, we've inherited 2 differences. So in the first case, where one of the 2 possible positions is different, it's called heterozygous, and this is the genotype. And at position 3, since both of the positions are different than the reference, this is a homozygous change. And so what we can actually do is we can only focus in on these positions to see what we expect, what is different, and how different is it. The rest of the positions, so example in position 4 or 6, we can look just to see what the reference has to know what our sample has. So this can help us simplify our focus even further. In fact, if we had a bunch of VCF files, and this is what this is, a variant call format file, we can identify patterns of how things are different, how often different samples and different individuals have differences in the reference, and that can help us. So if, for example, at position 10, we see a T, but 50% of the population has a T, probably nothing to worry about. But if in position 3, this is a rare variant, in fact, maybe 1 in 100,000 people have a G instead of a C, this could be potentially indicative of larger problems. So this is a small subset of what a VCF file might actually look at, lots of different positions, lots of different changes, and the evidence alongside. So a case study would be for sickle cell anemia. This is a disease often caused by a single change in the genetic code. This change is transcribed into the RNA message and eventually is written into the hemoglobin gene, which affects the shape and function of red blood cells. So at position 5,248,232 on the chromosome 11, you can see a T to an A change, which causes a protein change, which ends up with a structurally different shape of red blood cells. So normally red blood cells look like a donut with some, a film in the middle, and sickle cell blood cells have this characteristic thin curved shape. So what does this actually look like? So for a heterozygous patient on the left, we see a single change where half of the evidence shows the reference T, which is omitted, but is essentially the red elements. And on the right, we have a homozygous individual where every single read supports the claim that they have an A in this position instead of a T. Knowing these differences between not only the genotype, but the gene and the variant itself can aid clinical and diagnostic treatment options for patients or at the very least help individuals and patients know the root cause for different aspects of genetic disease. Excuse me. So once we have all this information, what can we do with it? So, excuse me, like in the case of sickle cell anemia, we talked about a rare single change disease, but we could also combine many samples into larger cohorts to provide general population information. How do variants distribute themselves across populations? How do different variants associate with different diseases like obesity or depression or more complex traits? Or you could focus in on cancer. How do our cancer cells genome differ from the comparable healthy tissue? Are there certain health outcomes influenced by different markers or genetic variants? And can any of this information be passed down through your children or from your parents based on these different risks? So that was the focus on DNA. We're going to switch a little bit into the RNA side of the message. So RNA sequencing allows us to investigate RNA molecules, and RNA is the messenger molecule that carries the information that is task-specific or cell-specific. So while all of our cells and all parts of our human body contain the same DNA, the RNA landscape or transcriptome can be vastly different between different parts of the body. So the messages that are being sent in the eye are drastically different than other tissues such as skin, liver, kidney, blood, brain. And it's important that our eyes and our lungs are doing different things, that our muscles and our spleen have different functions. So just a reminder, DNA and RNA are directly linked. It's all about which parts of the DNA are read that makes RNA so interesting. So RNA contains the message that is a subsection of DNA. So going back to this image, this is again the structure of genetic structure, how it is processed into pre-RNA. I think someone's microphone is on. And then further processed into messenger RNA. And so when we talk about RNA, we generally focus on messenger RNA, although this is not always true. And that normally mRNA, messenger RNA, excises and removes the introns in red. However, mistakes can happen, and there are genetic variants that can disrupt splicing, as we will soon see. So getting into the RNA sequencing steps, similar to DNA sequencing, we start off with a bunch of reads. We align those reads to a genome, and but the questions we're asking are slightly different. So while you can detect variants from RNA sequencing, in fact, I'd love to spend more time talking about that later, most of the focus goes towards changes in expression and differences in splicing. So, excuse me, expression is how much of the RNA aligns with different parts of the gene. So how much support do we have for using this exon compared to this exon? So we can see in this series that there is more usage of this compared to this, but even more so, you can compare how this gene is used compared to other genes for the different cell types. And then further, we have the different splicing behavior, which is how these different exons and these different blocks are combined. So we'll get into that more in a second, and we'll focus first on expression. So following the same model we've had on the last page, we have 3 exons and 2 introns, and we are going to remove all of the introns, like a perfect healthy splicing process, to read our DNA template into RNA, process it into messenger RNA, and then read that RNA into protein, which will go about and do things. However, if you're a brain cell or a blood cell, you might want to use this gene a little differently. So you can imagine a blood cell, for example, having to use more of this particular gene. So all it does is it uses the same DNA template, makes 3 copies of the RNA, processes these 3 copies, and you end up with 3 times the amount of protein. You have 3 times the RNA, 3 times the protein, and while that's not always true, they're generally, the more expression you have, the more protein you get. So that is expression, just counting how much of it you have. 1 copy versus 3 copies. However, you also have splicing. So the same cartoon, we've talked about cutting at these boundaries. So every red-blue boundary can cut and paste, and we can cut between exon 1 and intron 1 to remove intron 1, and then glue the exons back together. Here is an example where we cut everywhere we can, and we glue every exon back together, and we recycle the old introns. But again, what if you are a muscle cell that wants to do something different? You have a different use for this gene. So what you can do is you can change your splicing pattern. So using these exons as Lego blocks, you can create and interact and change the way you use them in different ways. And in this case, we can totally remove exon 2 to use the same genetic template to create a completely different protein which can interact with the cell and other proteins in different ways. But like I mentioned before, DNA and RNA are linked. And what if there's a mistake in the DNA? So in this example, there's a variant that messes up our DNA template, which is then written into our RNA message, which inhibits or prevents the cutting at this boundary. And instead, we do our normal exon 2, exon 3 cutting and splicing, but we end up with a completely different protein. In fact, this protein can be so messed up, or this message can be so messed up, that the protein is never made in the first place, or that the RNA itself is broken down before that even has a chance to happen. So enough with the cartoons, we can focus on some real life examples. So this is a particular gene, ACTN1, and the data comes from the Genotype Tissue Expression Project, GTEx, and we can see that different cell types use this gene differently. So this is a measure of expression on the y-axis and different cell types on the x-axis. And so we can see that our brain cells here in yellow have a drastically different usage from our artery and bladder cells here. And that's a good thing. I'm very happy that my brain and bladder do different things and that they're using the same gene in different ways. Similarly, as we can have expression pattern differences between cells, we could have splicing pattern differences between cells. So for the same gene, ACTN1, there are, I think, 20 odd splicing isoforms. So 20 different ways that you can use the same gene in different exon combinations to create slightly different proteins. So each tissue can use different splicing patterns to affect the proteins that they're making, which allow them to do different tasks within the cell. So getting into a even more focused version, we can focus on disease. So this is a case study in rare disease. Using RNA sequencing, we can improve diagnoses. So in this case, there's a patient with a particular mitochondrial disease, and this disease generally affects liver and skeletal muscle. And on the plots that we see, in plot B, we can see that for a given patient, we have a general distribution of how each gene in this sample is used compared to other samples in this cohort. So most of them are centered around 0. They don't deviate that much. However, we have one gene that not only has a drastically lower left of 0 expression pattern, but the pattern and use of this gene is drastically different than all the other samples that we in our cohort. And in fact, when we look at every single sample in our cohort, most of them use this gene with an expression value between 1,500 and 2,500 units. It doesn't particularly matter what they are. And our sample uses it closer to 100 or 200 units. And as a result, this is a good place to focus on the cause of this disease. So we can focus a little bit more about, instead of expression abnormalities, we can look at splicing abnormalities. So focusing here on the left, we can see between exons 5 and 6 in a normal sample, there is nothing particularly interesting. We splice between 5 and 6 as we're supposed to. However, in our patient, there is a change in splicing pattern. Instead of using this normal bridge 5 to 6, we have a new cryptic exon here in the middle with a new splicing pattern between it. In fact, you can look even closer about what is happening at this new splicing site, and there's a genetic variant that causes this part of the genome, or this part of the gene, to be included into the message, and this message includes some junk that causes the expression pattern that we saw in the last slide, that the message is so messed up, essentially, that there are mechanisms in the cell to recycle the message. They say, this can't be right, let's try again. But because of this genetic mistake, or this genetic variant, we see that this message cannot be recovered. So in summary for this part, RNA is carrying the message from DNA to the proteins, and each cell and tissue type use the same genetic information to accomplish different goals. This is what allows different tissues and different cell types to do different things, and that using RNA sequencing, we can dive into the differences here to allow researchers to continue digging into underlying diagnoses and pathways of disease. So again, a larger summary. At the genome level, we can identify coding variants that affect downstream proteins. Like in the case of sickle cell anemia, a single change affects the message, that message affects the protein, and that can impact the way that that disease is treated or studied. And at the RNA level, we can focus on expression and splicing patterns to identify why different parts of the body and different cell types might be using the DNA template in different ways. Finding whether or not these variants are important all depends on the accurate ability to sequence the DNA, sequence the RNA, and understanding this leads to downstream understanding of disease. So with that, I'd like to get into our first Q&A/intermission. Hopefully allow people to get a drink of water or ask any questions if you have them. Thanks.
Thank you, Nick, so much for the first part. Yes, and like Nick said, we're open for questions. If you have anything you want to ask Nick at this time about the slides or general genome biology, now is a good time.
I'd be happy to flip back and forth to whatever may or may not have been confusing.
Yeah, I have a question. In during your explanation, you mentioned something with the name of the isoform. May I ask what is isoform?
Sure. I apologize for getting too jargony. Isoform is a fancy word for a different version of the protein. So in this example, this could be the main way that this gene is being used. It uses this exon block, and so on. However, this could be a slightly or drastically different isoform, where instead you're using these building blocks to build your protein. So it just means a different way or a different version of that gene's protein or gene's message.
Thank you very much.
Mm-hmm.
If there are any other questions, you're welcome to unmute yourself or, oh, there's a question in the chat. Someone is asking, what is the difference between bulk tissue RNA-seq and single-cell RNA-seq? That's
a great question. So I didn't get into the differences between bulk and single-cell RNA sequencing, but essentially everything that I lay out here is, or that is represented in this slide, represents a collection of cells that are gathered from an individual that are then treated together to generate the RNA sequencing read. So for example, if we focus on this section here, we have muscle. So this would be in bulk RNA sequencing, I say for lack of a better word, a chunk of muscle cells collected and processed together to generate a kind of average understanding of what this group of cells is doing. However, single cell RNA sequencing focuses on the RNA within a single cell. And so this can help you differentiate differences within a broader cell type like muscle cell into individual pieces of the differences between cells. So for example, in your immune system, you have B cells and T cells. And if you had a blood sample, you might include both of those. But in a single cell RNA-seq experiment, you could individually study the differences between those two and identify what makes those different or how those are behaving differently. Does that answer the question? I know it was in the chat, but.
Sounds good. Nick, one question I was thinking about is you were talking about variations within genes, for example, with the sickle cell anemia, but there's so many stretches that are non-genes, right? So would variations in those areas also be able to cause disease?
Yeah, so let me see if I can find a good slide. So like I said, most of the genetic material in humans is non-coding. So in A, we could see a circle representing the genetic material for a gene, and B could be something intergenic between genes. And for a long time, people thought that regions like B were junk DNA, kind of not very important. However, since then, there's been a lot of discoveries about how genes are regulated. So not only are instructions about how to build a protein built into the gene, but instructions about how to read that gene are built into the upstream and downstream regions. So even though there are a lot of mechanisms for handling mistakes in genes or between genes, that there are certainly diseases and conditions where the underlying problem is a result of regulation, which generally could fall into regions like B, between genes. Does that answer the question?
Awesome, thank you. There are two more in the chat I'd like to read to you. First one is, could you explain once more the slide where you're showing differences between homozygous and heterozygous sequences for variant calling?
So in the cartoon version of it, because I've split it up into the genetic material inherited from one parent and the other, we can see that from one parent in position 2, we are inheriting a G, and parent number 2 is contributing a T at this position, and the reference has a T. So this means that in one of our 2 positions, we see what we expect, and the other, we see something new. And so this would be a heterozygous change. And in position 3, we expect to see a C. And in fact, both our parents contributed a G at this position. So we don't see the reference at all. We see the homozygous G at this location. For disease, or for a more realistic example, we don't know offhand which reads are from which parent. All we can see is the evidence and distribution. So we expect that in a homozygous patient, we would expect to see all of the same nucleotide at a particular position. And in a heterozygous individual, we would see a mix of the reference, red or unshown, and the variant A in green. And in fact, in rare disease, generally, you focus on homozygous cases because there's a lot of mechanisms for handling heterozygous variants and heterozygous mistakes. If there's a way for your body to recognize one version of your gene copy is quote unquote better than the other, then it can use that one instead to mitigate the downstream effects.
Awesome, thank you. And the final question, oh, there are actually 2 more questions, okay. So just to confirm, in simple words, gene expression is how much particular gene products are used by some cells, is that right?
That's exactly right. So gene expression really is useful when we start comparing different tissues or different samples across from each other. So whether or not in a single individual that you use 10 times more gene X than gene A is probably not as relevant as understanding that you use 10 times more of gene X in person 1 than gene X in person 2. Again, generally.
Awesome, thank you. And so finally, we have the question, who is the authority or what institution is it that publishes the human reference genome and how often is it updated?
That is a very good question. There is a lot of politics that go into that answer, unfortunately. And really, it kind of boils down to what people are using at the time. So currently, the human genome that most people use is version 38. However, you may have heard, which is a collection of samples from 20 individuals, I think in the United States, that their genetic information is included into the general reference. However, you might have heard of a recent development called the telomere to telomere project, which is the development of a new reference genome that uses newer technology and covers more of the human genome, which I didn't get into at all, but really goes and covers every single position in the human genome that is probably slowly but surely going to be incorporated into the new well-used reference. In fact, there's also another institution or another consortia developing a pan-genome project. Because the reference represents generally a small number of individuals or a single individual, that can't represent all of the deviation and variation within the human species. So the sad reality is that the majority of genomes that have been studied are of Caucasian descent. And so using this reference might not be as applicable to an individual who doesn't share the same genetic information, or not as much. And that gets into a lot of it, but essentially there are efforts to improve the reference, and depending on how good and how well established that reference project is, it gets accepted by computer scientists and bioinformaticians.